Advances in genetics. Science is continually advancing in the area of genetics, and there's been techniques which have enabled people to produce organisms with desirable traits. Selective breeding, cloning, and genetic engineering are three methods for developing organisms with these desirable traits. The process of selecting organisms with desired traits to be parents of the next generation is called selective breeding. Thousands of years ago, in what is now Mexico, the food that we call corn was developed this way. Every year, farmers saved seeds from the healthiest plants that produced the best food. And in the spring, they planted those seeds, and by repeating this process over and over, farmers developed plants that produce better corn. Selective breeding is done with plants and animals, such as these two selective breeding techniques. The technique of inbreeding involves crossing two individuals that have similar characteristics. For example, suppose a male and a female chicken are both plump and grow quickly. Their offspring will probably also have those desirable qualities. Inbred organisms have alleles that are very similar to those of their parents. Now, while this may increase production of larger chickens, it also increases the probability that organisms may inherit alleles that lead to genetic disorders. We've seen this several times with many breeds of dogs that have been bred and have now inherited hip problems. Hybridization is when breeders cross two genetically different individuals. The hybrid organism results in an offspring that has the best traits from both parents. For example, Macintosh apples were crossed with a Red Delicious apple to produce what is now called the Empire apple. This allowed us to have the taste of a Red Delicious apple, but the shelf life and resistance to insects that we would get from a Macintosh apple. There's a lot of research going into cloning now, and a clone is an organism that has exactly the same genes as the organism from which it was produced. It isn't hard to clone some kind of plant, such as a succulent, where you can just cut a stem from one plant and put the stem in soil, water it, and then soon you will have a whole new plant. This new plant is now genetically identical to the plant from which the stem was cut. However, scientists are also trying to clone animals, which is a much more complex method. This involves taking the nucleus of an animal's body cell and using that nucleus to produce a new animal. Now, with stem cell research on the rise, scientists are looking for other ways to clone not an entire organism, but parts of organisms that can be used to help in medicine, and such as growing organs for transplant patients. Geneticists have developed another powerful technique for producing organisms with desired traits, and this is in a process called genetic engineering. This is where genes from one organism are transferred into the DNA of another organism. One type of genetically engineered bacteria produces a protein called insulin. Injections of insulin are needed by people with diabetes. Now recall that bacteria have a single DNA molecule in their cytoplasm. Well, some bacteria cells also contain small circular pieces of DNA called plasmids. In this image, you can see how scientists insert the DNA for a human gene into the plasmid of bacteria. Once the gene is inserted into the plasmid, the bacteria cell and all of its offspring will now contain this human gene. As a result, the bacteria produces the protein that the human genes code for, and in this case, it's insulin. And because bacteria reproduces so quickly, large amounts of insulin can be produced in a short amount of time. Scientists can use other genetic engineering techniques to insert genes into animals as well. Genes have also been inserted into cells of plants such as tomatoes and rice. Now, someday, scientists hope to use something called gene therapy, which will involve inserting copies of genes directly into a person's cell. This will make it possible to use genetic engineering to correct some genetic disorders in humans. For example, researchers are looking for a way to treat hemophilia, which is a blood clotting disease. By replacing the defective allele on the X chromosome, the affected person's blood would then be able to clot normally. Because genetic engineering is so new, some people are very concerned with the long-term effects. So researchers are trying to be responsible by learning more about the effects of genetic engineering on plants, animals, and humans. Recent advances have enabled scientists to learn a great deal about human genetics. The Human Genome Project was an international scientific research project that had a goal to map out the DNA sequence of every gene in the human genome. Imagine trying to crack a code that is 6 billion letters long. Well, this is exactly what scientists did as a collaborative effort on the Human Genome Project, which was declared complete on April 14, 2003. Scientists have learned that the DNA of humans has at least 30,000 genes, and the average gene has about 3,000 bases. Scientists will one day know the DNA sequence of every human gene. Right now, even on your social media and the TV, you are probably currently seeing ads and commercials for DNA technology. Every day, people are sending in DNA samples from their saliva, which get broken down into small pieces or fragments. Selected fragments are used to produce what is called a DNA fingerprint. This DNA fingerprint helps identify people and show whether people are related. Some DNA fingerprinting can tie a person even to a scene of a crime, or can actually prevent the wrong person from going to jail. DNA records are being used to identify the bodies of unknown soldiers and civilians from past wars. However, like all technology, DNA fingerprinting has its limitations. It's very fragile, and the films produced can be difficult to read if the DNA samples are old. Better technology is getting produced every day, and DNA testing is taking less time and less money, and it is going to expand much more of what we know about human DNA and genes. Okay, it is time to show what you know.